Hi everyone, I'm Jade for anyone that doesn't know me. I'm working as a postdoc in Alex Wilgar's lab, and one of my main interests is in the rather novel combination of using TMS and fMRI. So it's a technique that's relatively still in its infancy, but it does have the potential to allow us to ask some pretty interesting questions about cognition in a causal framework. But as with all techniques that are in their infancy, it does require a little bit of methodological and theoretical development, which we are working on, and I'll talk to you a little bit about. But first, I'm going to tell you about all the positives and the exciting things that we can do with these techniques. So although this was covered in great detail, I'm just going to go through it very briefly. So historically, TMS is used to stimulate a region of interest with either excitatory or inhibitory stimulation, with the aim of disrupting that region's function and thus affecting performance on a task. Now, what we don't know with TMS is if you stimulate that region, is this also affecting interconnected networks or regions that are distal to that site of stimulation, and are they also really causally important for that task? Also, sometimes we don't get these behavioural effects with TMS, as this was touched upon, and we don't know if that's because we actually didn't hit our site of stimulation or something else went wrong. Now, with fMRI, which most of us are fairly familiar with, if we just think of like a standard task where you compare activation in different conditions, you get like a nice broad picture of what's happening in the brain. However, what we don't know is whether any of those regions are actually critically important for the task that you're doing. Perhaps none of them are actually causally involved, or maybe only one of them is. So that leads us on to the combination of using the two together. So with TMS fMRI, we can use it to stimulate one part of the brain and then look at modulated activation across the rest of the brain, the site that you've stimulated, potentially even subcortical structures. So that's really nice because, as this was touched upon, we don't really know about the neural effects of TMS, so it can teach us something about that. Also, it will hopefully tell us something about neural function. And then moreover, we can then relate these effects to participants' behaviour, so kind of this nice triad of looking at causal stimulation, how that's disrupted or modulated processing across the brain, and how that affects performance on a task. So I thought I'd just go through an early example, which I thought was quite nice, of combining these techniques together. So in Sakatel's work, they stimulated the right parietal cortex, and they found that participants, when they were doing a visual spatial task, this affected their performance, but it didn't during some control task. So that you can then say that this region's function is critically in visual spatial effort. At the same time, in the brain data, they saw that stimulation to the right parietal cortex specifically affected a distinct set of regions. And then this was also significantly correlated, the extent of deactivation, with how much their performance was impacted in the visual spatial task. So that's how we can combine all three things together, causally stimulation and modulation activation, and how this relates to behaviour. Now, our group is also interested in using TMS fMRI, and we started doing some work with it. However, instead of being interested in looking at overall activation levels, we're more interested in looking at how information is encoded across the system. And our interest for this stems mainly from some really interesting theoretical things that were brought up by DeWitt et al. In their paper titled "Is Neuroimaging Measuring Information in the Brain?" So here their main reasoning is that although as researchers, when we're putting people into an MRI or an MEG scanner, we think that we are decoding some type of information from them, we don't actually know whether this is truly information. It might not be useful, the information that we see encoded in the brain, to other regions in the brain. It might not be causally related, and it might not also be anything to do with their behaviour. So although we've done quite a few decoding studies together, for example, this work where we've shown that across this specific set of MD frontal parietal regions, you see encoding across the system of task-relevant information more strongly than you do of task-irrelevant. So these regions are adjusting their responses to code what's important for behaviour. What we don't know is if we actually disrupted one of these regions, would it impact on the coding that's contained in the other regions? So are they actually causally related, these codes? So we tested this with concurrent TMS fMRI. So we simulated the right dorsolateral prefrontal, which is one of the regions of this network, and we did that with both active or effective TMS and control or ineffective TMS. Participants did some type of selective attention task where they had to switch between attending to the colour or the form of a group of novel objects. And essentially the results are that when you disrupt or when you stimulate the right dorsolateral prefrontal with active TMS in comparison to control TMS, you see reduced coding of relevant information across the rest of the network, indicating that these codes are indeed causally related. You've now impacted one region and now the codes are affected elsewhere. So that in part kind of addresses Dewattel's reasoning in their paper, are these brain regions linked somehow? Is that information important for other brain regions? But next we want to move on to then see, does this impact participants' performance? So for example, when we see this reduced coding of relevant information, is this also correlated with the impact on their performance? So we're hoping to do this with the new concurrent TMS-MRI setup that we have at the CBU, where we also have an MR-compatible navigation system. So this is, again, also being touched upon. With the neuronavigation system, it's all about registering the structural data to the real-life person's head. So this is just an example of us mapping that in their structural data, so you just have to point to places on their head. And then the cool thing is now that we have this, the camera in the scanner, so we can actually map this, register them whilst they're in the scanner, and we can also get guided feedback on where to put the TMS coil. So the ultimate aim is to improve core accuracy and the target region that you're stimulating, which is much better if you're doing it in the scanner rather than outside the scanner, which is what we used to do. So the camera is emitting infrared light, and that's bouncing off of all of the little shiny balls on the tracker devices. So you have one for the participant's head, one for the TMS coil, and a pointer for the person's scalp. And then you use all of that for registration. Then next step is you get the person into the scanner. If registration is successful, then you can navigate the TMS coil to the right stimulation location that you selected in their structural data. And you get nice feedback about this whilst you're in the scanner room at the time, so you're more accurate about it, about both the location and the angle. Also, potentially, we can, though this is still working methods, we can receive that information about whether the coil has moved position or if we're exactly targeting the region we want to during the experiment itself, so whilst they're in there, because the camera stays in the scanner room the whole time. Also, we can potentially feed that information back to the participant to let them know if they've moved their head, so that they can move closer back to the coil if they want to. So we know that the exact location as well as the angle of simulation is really important from this nice work by Apitz et al. So they said that let's imagine 9 different zones that we're going to stimulate in the dorsolateral prefrontal cortex. All of them will count as DLPFC. Depending on the zone you stimulate, so if we look at zone 1 and zone 9, each row is a different network that you might be impacting on when you stimulate that zone. And also each column is a different angle that you are holding the TMS coil, and the color consists of how probable it is that you're going to impact on that network. So it doesn't only change the site that you stimulate when you move zone, obviously, but also the potential network. So in zone 1, if we just look at the row that shows the dorsal network, we can see that there's a probability that we might hit that region because there's a bit of green there, or that network. But if we look at zone 9, then there isn't really much probability that we can hit the dorsal network in that zone. Now, if we just focus on zone 1 and we look at the columns, so that's how we're holding the TMS coil, you can see that just going across any of those rows, say frontoparietal attention network, the colors are changing, indicating that you're more or less likely to hit the network depending on the angle that you hold the coil. So this is really important, and the navigation system in the scanner is going to help us improve that accuracy. However, we had other things before which are now also in improvement, which I wanted to tell you about. So you weren't always able to double check what was the site that you actually stimulated during your experiment, even with TMS-MRI, because in the old setup, the TMS coil was in between the MR head coil, so that picture on the left here, and this kind of hindered the MR excitation and readout of the actual site that you're stimulating. So you lost signal there completely. So you don't know if you hit the region you wanted to. However, with the new coils that we're getting in, that we purchased, these are dedicated to TMS-MRI, And this means that the MR coil with this new set is placed in between the TMS coil and the participant's head. So we avoid signal loss at the site of stimulation. It also allows us to increase the flexibility of where we put the coil. As you can see in the old setup, this is, it leads to some error because you have to kind of tilt the coil, etc., to get it into position. Hopefully also it increases participant comfort. However, what we don't know with this new setup is how much signal loss there is across the rest of the brain. So this is another development thing that we need to work out when we get to play with these coils. But we're getting 2 of them, so hopefully they'll increase signal from where both of the coils are located. Just the last thing to mention, because I'm running out of time. So one thing that happens when you zap people in the scanner is that you get an artifact that looks like that in your brain data, which is obviously not ideal, so these 4 pictures here. So how we used to deal with it is by interpolation. So that means removing the effector slice and replacing it with a mean of average of the preceding and the preceding slices. Then if you do that, you need to make sure that you're affecting a different slice on every volume, so you're not biasing your data and always removing the same slice from it. It's fine, but obviously you'll still have to account for these artifacts in your data, which you don't really want to have to do. So right now we're trying to develop here to see if we can stimulate between the gaps in acquisition instead and remove this problem whatsoever, so we don't have these artifacts in our data. So either this would be pulses delivered in the delay at the end of the TR, or potentially in the silence gaps between slices, which is the ideal case scenario, because not only are we avoiding those artifacts, but on top of that, we don't have to sync our TMS pulses to the TR. But there are downsides, like we have to have a longer TR, and that reduces the signal to noise ratio. So in summary, sorry if I've run over, but I hope everyone's excited now about TMS-MRI and having it here, and everyone wants to do it. But we just still have a little bit more methods development to do, as I said, improving the coil accuracy, which, as you all now know, is really important with TMS. Potentially feeding this back to participants. We need to pilot our new coils, and we need to figure out how to tightly control our timings to get the pulses in between the gaps in acquisition. Thank you, everyone.